Welcome to the software demonstration session. We have 4 software demos today. We had many more submissions than we could show today, so we apologize to all the ones that we had to delegate to the poster session. So please, everyone, join the poster session to look at the other amazing tools that are put out there. And this is a very traditional session that we have, so and it always attracts a lot of excitement. So that's very nice. Is there anything else? So yeah, so each speaker has a 10-minute time frame. So please stay within that limit. So 7-minute talking and 3-minute discussions, ideally. The first speaker that we have is Kerui Peng from the University of Southern California, and talking on pyTCR, which is a cloud-based platform for TCR-seq data analysis.
Hi everyone, my name is Kerui Peng, a second-year PhD student at the University of Southern California School of Pharmacy in Dr. Sergey Mangu's lab. And I'm honored and excited to be here to share our work, and thanks for offering me this opportunity. And as we gather virtually here today, we all agree that TCR repertoire analysis is really important in understanding the adaptive immune system and also in understanding its relationship in virus disease states. And most of us are familiar with the TCR analysis process, with the development of the TCR repertoire sequencing, TCR-seq, and development of awesome bioinformatics tools such as Mixer and VDJ tools and ImmuArc. We are able to capture the characteristics of TCR repertoires. However, we have observed barriers of the proper application of those bioinformatics tools among the bioinformatic researchers, and also especially with those biomedical researchers who have no or limited experience in the bioinformatics background, and then this really limits their ability to analyze the TCR-seq data in an efficient way. So our pyTCR is to aim to bridge the gap between those researchers and using these tools. So our pyTCR, we combine the TCR repertoire metrics and the statistical analysis and also the visualization at one place. And so here are the functionalities of our pyTCR. We include 6 different types of analysis, which include basic, clonality, overlap, VDJ gene usage, and diversity and motif analysis. And then next, I'll just quickly go through each type of analysis that we can do. So the basic analysis, we include all the basic metrics, and then here are the 2 graphs that we can plot for the clonal type count. And then here we use the COVID data that we got from the adaptive company. And then here are the COVID data that I categorized into 2 groups, which are hospitalized patient and non-hospitalized patient. And then next, for the clonality analysis, we include all the measurements that is important in this area. And then here we can show the bar plot for this relative abundance for the TCR repertoires. And then for the diversity analysis, we include all the important or common use metrics that we can show in the manuscript or the research papers. And then we have on the bottom part is a screenshot of what it looks like. So we have the code above, and then we have the result on the bottom part. And then here we show a plot that shows the difference between, for one of the metrics, that the difference between these 2 groups of patients. And then for the VDJ usage analysis, here is a heat map that we can plot from our tool. And then for the overlap analysis, we include all the important metrics, and also we can plot the heat map where the x-axis is the non-hospitalized patient, and then the y-axis is the hospitalized patient, and then there are differences between those overlap metrics. And then for the motif analysis, we are able to count the motifs. We can decide how long the motifs we want to look at, and also we can count on the amino acid motifs and also nucleotide motifs. And so what we are trying to emphasize here is that for our tool, for the traditional bioinformatics tool, it requires installation and some bioinformatics knowledge, or at least some experience, so they can use the command line and use write some code. And then but for us, these are not required. And also we are able to have a one-stop shop where we have the code and then the result and also the visualization part and also the statistical analysis at one place, instead of typing one type of code and then have the result at each single file. So I'm going to share my, I'm going to demonstrate how we are going to use this. So for our tool, we provide this in two different formats. One is in the Google Colab. For the people who are familiar with the Google Colab, it's relatively easy to use and it's free to use for all the Google users. So I have 7 different types of notebooks here, and also we also provide this in the GitHub page for other people who are familiar with the bioinformatics, and then they can just use the get a copy of our Jupyter notebook from the GitHub page, and then can run on their local computers. So here, what they need to do is just click one cell, and then they will import the Python libraries. And then they have 2 options. Actually, they have 3. One is to upload the data file from the Google Drive, where if each one of us who have a Google account, then we can have 50 gigabytes of the Google Drive, and then which we just need to mount the Google Drive to this and then give authorized Google Drive authorized to the Google Colab. And then next, I'm just going to authorize it right now. So it should be fast. After it's authorized, then it can read the file from the file we already uploaded to the Google Drive, and here is a template that we have. And then the second option is people can upload their file from the local computer, and the third option is actually they can run this on their local computer as well. And then we create this standardized format. And then people can just simply click the link, and then the result will just come out really easily. So it's all at one place, and if people want to save the file, it's relatively easy as well. And also the plot is easy to generate and it's easy to see, and it's really transparent as people can see the code and adjust as they want. Sorry, I'm going to go back to the slides now. So as a conclusion, as our notebook, it includes all the functionalities that the existing tools have, and also we provide more. So it is a comprehensive cloud-based method for TCR repertoire analysis and visualization, and it is a one-stop shop for the biomedical researchers who has limited bioinformatics background, but they have big data for analysis. And our tool offers a broader and more powerful functions in the TCR repertoire research. And I just want to thank my co-mentors, Dr. Sergey Mengu and Dr. Huda Lachka, for offering me the opportunity and giving me guidance, and also the members in those 2 labs. And that's conclude my talk, and thank you for the opportunity. I'm willing to take any questions right now, and also I have a poster session tomorrow. If you have more questions, you can ask me at that time too. Thank you.
Thanks so much for the wonderful talk. Do we have questions? I don't see any right now, so maybe I will ask a question. Maybe you have mentioned that, but how does your tool platform compare to other platforms, such as, for example, ImmuneArc or BDJ tools or things like that?
Yeah, I think so. So I think we compared the functionalities between those two platforms, and there are differences between those two, such as there are some functionalities are not existing in one tool but existing in other tool. So we combined all those functionalities together in our tool, and also this work is still going on, and we are working with Dr. Mike Shugai, who is the developer of the mixer and also of the VDJ tools, and we are collaborating with him, and we will provide more functionalities that we can put in our platform. And I think for our platform, the important thing that we want to emphasize is that it's pretty transparent, and I guess in our lab we like this Jupyter notebook form, where we can see the code, and also we can see the visualization. We can see everything is very transparent. And yeah, that's the motivation for this work is that we have seen some when biomedical researchers who have limited background, when they want to use these tools, sometimes they have a hard time to learn, and sometimes they have a hard time to use. And also when they type a code, sometimes they don't know what filtering, I guess what is behind the tools. It's like a black box sometimes.
Yeah, that's good. So one question, and then we have to move on in the interest of time, from George Blank. He asks, is there an advantage of Google Colab over Docker and creating a container with for pyTCR? What is the latter option considered or compared?
I guess so far it's not. I guess I haven't used this Docker before, so far we only have this in the Google Colab and also in the the GitHub page. But I will definitely look into this since we are still developing and trying to make this better. Thank you for the suggestion.
Thanks a lot. So then we move on to the next speaker. Thanks so much.